last speaker is going to be Professor Maria Luisa Serrano Garcia. She's from Venezuela. Maria Luisa is a full professor of the School of Pharmacy from Universidad Central de Venezuela. She currently leads the medicinal chemistry unit. She's also an external researcher at the Tropical Medicine Institute, and her research focuses on protein homology modeling, molecular dynamics, and computer-aided drug design with applications on relevant targets in viral and tropical diseases. She's an author of more than 25 scientific articles. She has directed the research of 5 PhD students, several specialists, and undergraduate students. The title of her talk is In Silico Strategies, Key Methods for Drug Discovery. So Maria Luisa, go ahead with your talk.
Okay, thank you very much, Ariela. I'm really, really happy to be here. I think we all are very happy to share our work with you. I just want to check if you can see the presentation.
Yes, we can see it, but you have to share, the full screen. Now it's okay.
Now it's okay. Okay. Well, let's start a little bit. This is my the title of my talk, In Silico Strategies and Key Methods for Drug Discovery. Of course, I know everyone knows a lot about that. But first, I would like to tell you who we are. We are a group that works in medicinal chemistry. We work at the School of Pharmacy at the Universidad Central de Venezuela. But as medicinal chemists, we strong believe in collaborations. So we have very strong collaborations with different researchers and groups. One in the Instituto de Medicina Tropical, I work for them or with them very strongly, with Dr. Noya in that institute. And we also have a very strong collaborations with 2 groups, with Mariana Hidalgo, as a parasitologist, and Flor Pujol and Héctor Rangel at the Instituto Venezolano de Investigaciones Científicas. And part of the work I'm going to share with you is in collaboration with them. So mainly our motivation is to work in neglected diseases, not only because they affect a huge amount of people worldwide, it affects more than 20% of the world population, but because most of that people is in our countries, and most of them have or are affected by at least 5 of these neglected diseases: malaria, leishmaniasis, Chagas, HIV. But the other concern is when you think about the money that you have to do research, you can find that is very, very low, the spending of that you have to develop this research. So it is very important to work in those kind of neglected diseases for us. Our strategy, it's a very common strategy, but we usually start with drug target identification. Most of the times, this is in collaborations of one of our partners that do the identification and characterizations of new targets. And then in our lab, we usually do homology modeling of these potential drug targets. Because as you probably know, it's very few the information that you can find in the PDB for structural targets for neglected diseases. And then we use those structures to drug design, virtual screening, or to explain some biological data that we have in our hands. And finally, we go back again to our collaborators to do the biological assays of the compounds that we have identified. So I would like to share with you just 3 different projects that we carry out the past years, just to show you that our main interest is what is the question that we have in mind to design the project and to decide what is the in silico approach that we have to use. So the first case we're going to discuss is about malaria. We are very interested in Plasmodium vivax malaria, not only because it affects around 200 million people around the world worldwide, is also the predominant parasite in Latin America, but also is the main parasite in Venezuela. Of course, for this disease, there is a combination of drugs that are currently used. There is primaquine and chloroquine. They are both used as a combination to treat the disease, but there has emerged drug-resistant strains for this. So it is very important to continue the research, to find new targets and to find new drugs. But particularly for Plasmodium vivax, it's very difficult to study because it's not easy to cultivate that parasite in the lab. So the information must be about the proteome have to be studied directly from patients. Around in 211, Archaya group do a proteomic study about this parasite, and there they identified potential new targets. One of those was Helicase, an RNA dead box Helicase. So at the parasitology lab in the EVIC, they identified this Helicase and a study that sequence and characterize them by tandem mass spectrometry. These helicases are very, very important because they bind and remodel RNA or RNA protein complexes in ATP-dependent fashion. And they could be selective targets because its function is essential for parasite viability and differs sufficiently from the one that are present in the mammalian human host. To begin with this part of the talk, I would like to show you a little bit about the conformational changes that suffers these helicases in the catalytic cycle. At the beginning, these helicases have 2 domains, and they can be in the open state in the absence of ATP or RNA. This open state is the inactive state. And then when it bounds to RNA and ATP in a collaborative way, they close together and get to the closed state, that is the active state of this helicase. So we decide to have a general strategy, and we initially, after we have the sequence and the characterization of these helicase, we generate homology models, and do it molecular dynamic simulation for stability assessment, the classical molecular dynamic simulations. And we build the 2 models. We build the closed state model and the open state model of this enzyme. The closed state, we use those after analyzing the RNA binding site. We use this closed state or active state for virtual screening of a library of compounds, select those compounds, and do biological evaluation. And we also use this closed state to design and to synthesize that after peptides that can also bind this helicase with what were our thoughts that is this peptide can bind the open state so they can prevent the closure of the helicase. We also do these peptides, we synthesize those peptides, study a little bit of docking with the open state, and go to biological evaluation. I'm not going to talk about this part, just of the virtual screening part. So we have both the closed state and the open state, active and inactive ways of the protein. We validate our models as usually you do. But where we're planning to do our virtual screening approach, we decide to use the two helicases, the P-Vivax helicase and the human helicase, just to exploit the differences. Sorry? Okay.
No, go ahead. It's fine.
Okay, go ahead. Okay. So we compare the two helicases just to be sure that we can do a virtual screening and try to select with selectivity for the P-Vivax helicases, exploiting the differences that we found in the active site, the RNA active site, for those helicases. So on the virtual screening, what we did was to use the malaria box. This is a box of compounds that was designed in medicine for malaria venture. The advantage that we have here is that malaria box, you have the virtual library that is compounded of 200 drug-like chemotypes and 200 probe-like chemotypes. But the advantage is that at the end of the screening, you can ask for the compounds, they send us to you, and you can do your biological evaluation. So, okay. Then we screen by molecular docking. We use two different approaches. We use autodock and we use also glide to do the virtual screening and exploit the differences. You can see here in the closed state of the protein, the differences of the two enzymes. So you can find when you do your virtual screening, a selectivity between the two helicases, so you can select the one that is bind with high affinity to your protein of interest, in this case, the P-Vivax DAG box helicase. So in that way, we select 10 compounds and we go to the biological evaluation of these compounds, doing cell culture from the synchronization of Plasmodium joelli lethal strains. I want to explain this a little bit. We use these strains because, as I told you, you cannot have the parasite Plasmodium vivax in the lab. You cannot cultivate that parasite. You have to get it from patients. But when you compare the amino acid sequence of Plasmodium joelli, is 90% of identity with the Plasmodium vivax strain, and also there is a 100% of conservation in the active site. That's one reason. And the other reason is that the antibody that you use to study the Plasmodium vivax protein also recognize these Plasmodium joelli helicase and not Plasmodium falciparum helicase. So you can use that as a model to the preliminary biological evaluation. So then what we do is to follow up the development of the different parasitic forms with the compounds of interest. In these graphics, we can see the evolution with different concentrations of the drug. This is just an example of one of these drugs. The concentration of these drugs, artemisinin as a reference, and chloroquine as a reference, and the solvent. It's important just to be clear with the colors of the graphs that the merozoite, that is the form that invades our red blood cells, first it develops as trophozoites, then they go to the ring form, and finally to the schizons form. So this is the last part of the evolution of the parasite inside the red blood. So what you can see here is that our compounds, so this is one of the best compounds at a micromolar dose at 20 hours, it behaves like artemisinin and like chloroquine. I mean, they stop the growing of the parasite. They don't develop to schizons. So there is no yellow form expression here. They just stop in the ring state of the parasite. So this is very, very encouraging. These we think they are schizonticidal type of compounds like artemisinin and chloroquine. And now we are doing just molecular biology work with the expression of the proteins and continue this work. This is carried on also. Well, the second example I would like to share is our case of studies with COVID-19. Last year was really very important for the scientific community. It makes a lot of pressure in the scientific community trying to search for new compounds. And we were working in viruses with the group in the EDIC Institute for a few times. We worked in hepatitis C and we worked also in HIV. So last year we have a few occasions in SARS-CoV-2 virus. I'm not going to talk a lot of that, just a brief introduction. This COVID-19 is caused by the SARS-CoV-2 virus. That is a coronaviridae family, and it's a beta coronavirus. When you look at the great big genome that you have, this virus have, there are a lot of proteins that could be potential drug targets. Some of those are more relevant because they play principal roles in the viral life cycle, and because also you don't have human proteins homologs like the one that we're going to talk in this example, are those protease, the papain-like protease and the main protease of this virus. But what is important here also that there are not only these two proteases that are involved in virus replication, there are also a human host protease that is involved in this virus. So that is the human host transmembrane type 2 protease. For this one, there was no crystal structure, so we modeled the protein to use in the work. This time, we asked us a different question. Thinking about the repurposing of drugs, there was a clinical study that was carried out last year about famotidine. That is a drug that is a H2 blocker and an antagonist of the histamine 2 receptor that is used for gastrointestinal efflux, and is approved to use for that. But also this drug, famotidine, is also, it has been proposed as an HIV-2 inhibitor of the main protease. So we think, well, let's see, it's not clear if this famotidine could be an inhibitor of the SARS-CoV-2 protease. So we decided to study the binding of this drug with not only the transmembrane protease type 2, but also with the two main proteins of this virus that are involved in the virus, in the replication of the virus, and the HIV protease. So we built a small library of compounds, always using reference compounds that we know that also binds to the enzymes. And we use for that autodoc, and then the final analysis as everybody usually do. But we also, for a couple of drugs, of this drug and another couple of drugs, we calculate physicochemical properties that associate with the ATME properties and do also that kind of analysis. Okay, so what we found is that really famotidine could bind to all the different proteases, but with really low binding affinity. We can see, for example, in the human protease, that the binding energy is really low, well, relatively low. When you compare with the other inhibitors, also is the same thing that we can observe with the viral papain-like protease that is also binding low binding free energy. When you compare that with ribavirin, for example, that is a well-known inhibitor of this protease. And the same thing happened with the other two enzymes. In this case, that HIV is not shown, but with the viral main protease, you can also see that is also the lower binding free energy. But although there is a low binding free energy, we can see anyway that they can bind all the three proteases. So it could be used as a drug, but probably in combination with another drug, okay? So the other thing that we usually do in our medicinal chemistry studies is that we calculate the pharmacokinetic parameters. These pharmacokinetic parameters are well known, and the parameters that are associated with the Lipinski rules. So like lipophilicity, number of insaturations, and the size of the molecules, etc. And in these radar plots, we can see that everything is inside this pink area is all those drugs that have good properties to be oral viability. So what we did is we took a number of different compounds that are well known as antiviral drugs that were previously evaluated against SARS-CoV-2 virus, and we calculate those properties and compare them. What we found is that famotidine, as you can see, the red line, is that it could be as good as ribavirin, that is another drug. They are all good for oral viability, and also that famotidine is very polar. So this is important because it also can be used to use it intravenously. You can inject the drug so it can reach the lungs. That is very important for this disease because, at the latest state of the disease, the replication usually occurs in the lungs. So it's important that you can reach the tissue because of this high polar characteristic. So what we propose in this work, we can read it, all of it, is that it can be used with hydroxychloroquine, and we are just trying to see what happens with the clinical trials that were carried out last year. Well, The other case of study I would like to share is a project that is still ongoing, and is a very different approach of in silico methods. We are trying to develop SARS-CoV-2 serological testing, because as I told you, we have also a wet chemist lab, and we do peptide synthesis. So this is a project that is in collaboration with Dr. Oscar Noya groups in the Instituto de Medicina Tropical. So what we want to develop is a test. Then you can use synthetic peptides instead of the whole antigen to catch the antibodies that are in the sera of the patients. When you develop those tests, you can use all the antigen, you can fix them in the test, and you can catch the antibody that are in the blood of the patients that have COVID-19. But you can also, instead of using the whole antigen, the whole protein, you can use a fragment of that, because just the antigen just bound to the antibody with a small region that is called the epitope, and you can synthesize those epitopes and try to catch all these antibodies that are in the serum of the patients. So in this type of projects, you can use different strategies. You can use just sequence analysis if you don't have any information about this structure, but you can also use an antigen structure analysis, because you can look into the antigen, the regions that are exposed to solvent, and there select the regions you want to synthesize. But more of that, if you have an antibody-antigen complex, you can analyze the region of contact and select that region to be synthesized like a fragment, like a peptide. Okay. In SARS-CoV, with the virus SARS-CoV-2, there are a lot of information, a lot of crystal information that was developed last year. So we have a lot of structural information, not only about the proteins. We we analyze different proteins that are important as antigens for this virus. And we also analyze protein complexes, particularly in the case of the spike protein. And we made a selection after analyzing these proteins, looking for the regions that are called B-cell immunodominant regions. We select those regions after analyzing just not only the sequence for linear epitopes, but also analyzing the structures and the protein complexes. We select around 206 peptides to be synthesized. And as the chemical synthesis is very, very expensive for us, so we use a very particular technique. That is, we synthesize those peptides with that peptide library over a cellulose membrane. So you link the peptide to a sheet of paper of a cellular membrane. You build over all the peptides like it's spots. This is well known as the spot technique of peptide synthesis. So you have a little membrane that is usually around 15 per 15 centimeters. You have all your peptides linked to that membrane, and then you do a immunoassay by using this small membrane, and you expose the membrane to the sera of the patients that have the antibodies because they were sick with this COVID-19 sickness. So then we use pool of sera to do that, and what you can see in the right is the results of that assay. And in each spot you have a peptide, and when you have a strong signal, black signal or gray signal, you can see that these peptides are recognized. So there is a very interesting way of selecting the peptides, analyzing the structures and analyzing the protein complexes to select those peptides. So these peptides that are highly recognized here by the antibodies, the EgG antibodies and the EgM antibodies, are now going to be synthesized in more quantity by solid phase synthesis and being evaluated by any other of the well-known techniques. This is also was published last year, and you can find the information that you want there. So I just want to thank you, all the people that works together. Luisa Serrano was my PhD student that was working with malaria, and all the work in COVID was with Dr. Pujol and Hector Rangel at the INVIC Institute, and the last part with the group of Dr. Noya. And I want just to share with you just final thoughts. It's very important for us to be together. We are women that work. I have a big family. This is all my kids. I have 4 kids. And this is my second family. Then we have to work hard, but we also have to enjoy science and enjoy life. Okay, thank you very much for your attention. And if you have any questions, I will be very glad to answer them.
Thanks, Maria Luisa, for your talk. We have a question here. Could you clarify a little bit how is the initial protein identification and characterization step that you perform? This question is from Laura.
In the, I think that she is trying to refer in the malaria work.
I think so, the first part of your talk.
Okay. This protein was characterized by Dr. Hidalgo. She is a molecular biologist and a parasitologist in the INVIC Institute. So they purified the enzyme with a molecular antibody, and then she just picked the protein and then isolated the protein and characterize the protein and know the sequence with tandem mass spectrometry. So we have a novel sequence, but of course it's a novel sequence, so we don't have any structure, and that's why we model this sequence and start our drug discovery project.
Okay, Maria Luisa.
I hope that is answered.
Okay, thank you so much. So